So today we are going to discuss about control and coordination. So you know, control and coordination is being controlled by brain itself, right? So what are the functions of brain? And brain, you know, consists of neurons, right? Neurons are somatic cells basically. So neurons are the somatic cells, and specifically if I ask what is neurons, तो आप क्या बोलोगे? Neurons are highly specialized cell, right? Highly specialized cell. And if I talk about the function, it will receive and transmit. Receive and transmit what? Signals and impulse. That means function wise if I talk, so it will receive and transmits the impulse and signals in the form of electrical charge, which is negatively charged. Okay? Now if I talk about the chemicals which are involved in this process, the entirely chemicals are called neurotransmitters. Neurotransmitters. That means neurotransmitters are the chemicals which are helping the neurons to receive and to propagate the impulse or signals. Clear? Okay. Now we will be discussing about the classification of nervous system. As we know, the system which is related to the neurons, that means taking decisions, reflex actions, is combinedly called nervous system. Like respiratory system, the system which is related to the gaseous exchange. Excretory system, the system which is related to removal of body waste and excess waste from the body. Similarly, the nervous system, you can say, nervous system is broadly classified into two parts. Okay, first one is your CNS and second one is your PNS. Clear? So CNS stands for Central Nervous System and PNS stands for Peripheral Nervous System. Clear? So specifically if we talk about the central nervous system, which consists of brain plus spinal cord, right? So your spinal cord and brain together form your central nervous system, which perform most of the intelligent tasks of the neurons. That means receiving, taking decisions, performing several actions like reflex actions, how to stimulate how to respond to the stimulus. These kinds of action is being controlled by your CNS, and additionally it is being supported by peripheral nervous system. Clear? Now specifically if we talk about the classification of peripheral nervous system, it is again of two types. One is your somatic and autonomic. Okay, somatic and autonomic. So what do you mean by somatic? Somatic, simply you can say that the nervous system which is connected to muscles, somehow which is connected to muscles, and specifically if we talk, it is also again divided into two parts. One is your afferent, or you can say that sensory, okay, and second one is your efferent neurons, which is known as your motor. Okay, let me give you a brief idea about the sensory and motor. Afferent term, in the sense, you should remember always, afferent means two words. Afferent means two words. In case of excretory system also we have learned that afferent arteriole and efferent arteriole in the vacuum capsule, right? That means here I can say that afferent neurons, that means the neurons which transmits the impulse or signals from tissues to CNS. Clear? Afferent neurons are the neurons which are responsible for transmitting the information or signals or impulse from tissue to CNS. That is known as your afferent or otherwise known as sensory. Receptors also you can say that receptor. That means that receive the information. We have 5 sense organs. All the 5 sense organs receives different types of information from the surrounding by the help of sensory neurons or afferent neurons. Clear? Now secondly we will be discussing about the efferent one. Obviously vice versa. Efferent means away. That means which transmits the message or impulse from away from the CNS. That means same, after receiving the impulse, the CNS has to take some decisions. After confining a decision, it will again pass back or reverse the message from CNS to tissue or specific organ. Let's suppose I saw a snake. So I got the information by help of afferent neuron from the eye, which passed the information to the brain, and my brain decides to run away. I should run away. So that decision will be carried forward by efferent neurons, which is specifically transfer the information to the leg, and my leg muscles will start to work and I can start running. That work is being done by efferent neurons or motor neurons. Clear? Now we will be discussing about autonomic one. Autonomic is also being divided into 2 types. Okay? First one is sympathetic and parasympathetic. Clear? So first one is sympathetic, second one is parasympathetic. Sympathetic in the sense how you are reacting towards a stressful situation or to a situation where your neurons are very much excited. That state is otherwise known as fight and flight stage. It is otherwise known as fight and flight. Clear? And specifically if we talk about parasympathetic neurons, which is known as resting. Okay, sympathetic can be also called arousal, which increase your arousal towards something. That is known as sympathetic, and parasympathetic is the reverse of sympathetic, which is known as resting. So this is the broad classification. Again I am discussing. Nervous system is being divided into CNS and PNS, central nervous system and peripheral nervous system. Central nervous system, which consists of brain and spinal cord, and peripheral nervous system is further divided into somatic nervous system and autonomic nervous system. Okay, somatic nervous system is further divided into afferent or sensory and efferent or motor, and autonomic is being divided into sympathetic and parasympathetic. Okay, now we will be discussing about the structure of neurons. The structure of neurons. Okay. So first of all, let me introduce. There are total 4 components. How many components? 4 components. What are they? So specifically, the first one will be cell body. Okay. Then dendrite. Okay. The third one will be axon. And the fourth one will be axon terminal. Axon terminal. Okay. So let's discuss one by one. Okay. So let's have a clear idea about the diagram. So if I will draw the diagrammatic view, somehow it will look like Okay. So here I can clearly visualize the central part, which is the nucleus. It is the nucleus. Okay. Then this part is your dendrites, and this part is your cell body. The internal part is your cell body, which consists of cytoplasm, unlike other cells. Like cell consists of nucleus and cytoplasm obviously, and cell membrane is also there. So cytoplasm, along with that few organelles and few granules. And the granules are specifically called Nissel's granules. It is called Nissel's granules. Now let me remind you, what do you mean by Nissel's granule? What is the functions of Nissel's granules? Nissel's granules helps to synthesis protein by the help of ribosome itself. Okay? Nissel's granules helps to formation by synthesis of protein by association of ribosome. That means function wise, Nissel's granules helps to produce protein. Clear? Okay. Now these granules are somehow you can say that. Okay. So after secondly we will be discussing about the dendrites. So dendrites are branched short strands which receives, the functions of dendrite is to receive the impulse from axon. The function wise it receives the impulse from axon. Okay? Okay. And specifically count wise if you talk, there are around 4 lakhs of dendrites can be available in your human body. Okay? Now we will be discussing about your axon. Cell body is over, dendrite is over, now we will be discussing about the axon. So axon is the long strand which receives the impulse from cell body. That means if I talk about the flow of impulse, it will be like cell body which transfer the impulse to axon, which transfer the impulse to axon terminal, and which again to the synaptically shaped nerves to dendrites. Okay, the region between axon terminal and dendrite will be called synaptic cleft. Synaptic cleft. These are the nerves. Okay, these are the nerves. That means likewise another neuron should be here, from which the axon terminal will be attached to the next dendrite of next neuron. Likewise, your impulse can transfer contact from one neuron to other neuron. That means throughout the body, it is flowing like in current or a negative charge. Clear? Now we are discussing about this part. Okay. Okay. Now we will be discussing about these parts. Specifically, these are the myelin sheath. These are the myelin sheath. What is the myelin sheath? Myelin sheaths are made up of lipids. Remember, myelin sheaths are made up of lipid. Now, what is the benefit and what is the role of lipids here? As we know that electrical impulses are negatively charged, they need to be carried forward or conducted in a uniform direction. So, lipid helps us to provide the insulation to the conduction channel. That means the myelin sheath will help us to maintain the insulating factor. That means it will act as an insulating barrier which can allow the flow of current in a particular direction. Okay, so these myelin sheaths can be contributed by few cells. These cells are called scavenger cells. Okay, which is a part of, remember, PNS. Scavenger cells are the part of PNS. In case of CNS, scavenger cells are not present. Instead of scavenger cells, there are oligodendrogolia is present in case of CNS. In case of CNS, these scavenger cells are absent, and instead of scavenger cells, oligodendrogolia is present. Clear? Now, look at the structure over here. Okay, as I said, these layers are acting like an insulating barrier. That means whenever there is a requirement to exchange ions or any kind of impulse, it is required, that can be done through this known as nodes of Ranvier. Nodes of Ranvier. That means any kind of exchange between the cell and surrounding is possible at the point by nodes of Ranvier. That means the diffusion is possible in nodes of Ranvier. Clear? And additional one thing I need to add here, that here centrosome is absent. In case of neuron specifically, centrosomes are absent. Okay? Centrosomes absent. Why? Because they lack the power of division. Due to absence of cell division, they lack centrosome as well as centriole. Clear? So, here you can say that centrosome and centriole is absent due to lack of cell division. So, this is all about your structure of neurons. So, let me give you a brief idea again. So, it is made up of 4 components: cell body, then dendrites, then axon, and last one is your axon terminal. Clear? Okay. Now, we will be discussing about the types of neurons. Here also we have discussed types. First one is your afferent neuron and efferent neuron. Yes, as I said, from tissues to CNS, and this one is from CNS to tissues. Okay, so broadly if I classify this, we can classify depending upon their structure, depending upon their function, and depending upon the presence of myelin sheath. Based upon these 3 characters, we can divide them into different categories. Okay, so first of all, let us discuss about the types of structure. So according to the structure, we can divide them or classify them into 3 categories. One is your unipolar neuron. Second is your bipolar neuron. Third is your multipolar neuron. Okay, so according to the structure, I can classify the neurons into 3 categories. So what are they? Let's discuss. First, unipolar. The name suggests itself. Uni stands for 1. Okay, it is very easy. If I line over here, when a neuron contains only 1 axon. Simple. That means the neuron is having only one axon, that means there is no dendrites. There is no dendrites. These types of neurons are called unipolar. Structure wise, they have look like this, somehow look like this. That means only axon is here. Okay? There is no dendrons present. Only axon. So this kind of structure is known as unipolar. Now question arises, where this kind of neurons can be found? These kinds of neurons can be found in case of invertebrates. In the organisms like invertebrates, we can found unipolar neurons. Clear? Second, we can move to bipolar. Are the names of just bi? That means two. Okay? So this is just modification. That means consists of two axons in a reverse manner. In a reverse manner, these kinds of neuron are called bipolar, which can be found in case of retina of eye. Retina of eye. We can found bipolar neurons. Okay? And last one is your multipolar. Multipolar, that means where the dendrites of one neuron can bind to several axons of different neurons. Where different types of dendrites arises from the cytal region. From the cytal. Okay? From the cytal, there are different types of collaterals and different types of new dendrites are arising which can bound to different axons. Okay? Which is found typically in CNS. The neurons which are present in CNS are multipolar in nature because they have to receive the impulse from various receptors. So the dendrite has to connect it to different axons. So this where dendrites connected to different axons, this kind of structure is known as multipolar neurons. Clear? Okay. Then functions. Okay. Then we'll be coming to based on functions. Based on functions. So based on functions also there are three types. Already we have discussed two from them. One is your afferent neuron. Second one is your efferent neuron. And last one is your interneurons. Okay. Afferent neurons already we have discussed. Which transmits the information from tissue to CNS. Efferent is from CNS to tissue. Now come to interneurons. Interneurons are the neurons which are specifically or specializedly present in brain or CNS. Interneurons are the neurons. Interneurons is the third type of neuron according to the functions. As I discussed, afferent neurons and interneurons. Interneurons are the neurons which are specifically found in CNS. That means all the coordinated activity, controlling nature is being conducted by interneurons. That we can alternatively say that afferent neuron and efferent neurons are connected to interneurons only. Whatever the impulse is being transferred or signaled, that is by the help of interneurons only. That is the control system of brain. Okay. Now, last we will be discussing about the according to their presence of myelin sheath. Myelin sheath. Again, it is being divided into two types. If the myelin sheath is present, that will be called regulated. And if it is myelin sheath are absent, simply you can say that non-regulated. Okay? If myelin sheath is present, then it is called Modulated neurons. If myelin sheath is not present, we can say that it is non-modulated. Okay, and non-modulateds are somehow we can say that found in PNS, and these are say found in CNS. Modulateds are found in CNS and non-modulated are found in PNS. Already I have discussed the functions of myelin sheath, which is act as an insulator. Which is act as an insulator and made up of lipids. Clear? Okay. Now we'll be discussing about the process of a mechanism, how impulse is transforming or how impulse is being how impulse are carrying forwarding from one neuron to other neuron. Okay? Okay. So for that, the mechanism is being processed with 3 stages. The stages will be like polarization, which is otherwise known as resting potential. Second one is your depolarization, which is known as action potential. And last, third one is known as your repolarization. Okay, polarization, depolarization and repolarization. Let's discuss one by one. First one is your polarization, which is otherwise known as resting potential. Resting potential. Okay, so what happens, as we know, electrical signals which are conducted from negatively to positive, because negative charge is being attracted towards positive, right? So whenever a cell is excited or receives an impulse, it receives a negative charge inside the cell. I am talking about inside the cell and outside the cell. That means this is the surrounding and this is the inside of a cell. We will be discussing about different components and electrolytes which helping us to perform this activity. So one is your sodium and potassium. These two elements take some vital role to exchange impulse from neurons to neurons or neurons to any other component. Okay, so specifically if we talk about the polarization case, remember the inside cell will be negatively charged. Inside cell will be negatively charged, and the concentration of potassium will be high inside the cell. If the concentration of potassium will be high inside the cell, automatically the concentration of potassium will be low inside the cell. Outside the cell. Right? If the concentration of potassium will be high inside the cell, that means by default the potassium concentration will be low outside the cell. And reversely, the concentration of sodium will be low inside the cell and the concentration of sodium will high outside the cell, which by default can create the concentration gradient. That means there is a difference between the concentration. Potassium is exhibiting high concentration inside the cell and low concentration outside. Same, sodium is concentrating low concentration inside the cell and high concentration outside the cell, which automatically creates a concentration gradient. That means the sodium and potassium pump will initiate. Sodium and potassium pump will initiate. Due to presence of negative charge and potential difference, this phase is known as resting phase, which will incur a negative charge. And this condition is known as resting potential. And this stage is known as polarized by polarization due to occurrence of polarized cell. Polarized cell in the sense negative charge inside the cell, high potassium concentration and low sodium concentration. Clear? Now we will be moving towards the second stage, which is known as depolarization stage. In depolarization stage, what will happen? Obviously, the substance from higher concentration will move to lower concentration. That means the sodium-potassium pump will allow the flow of sodium from outside to inside and potassium from inside to outside. But here the force of potassium is greater than sodium. That means more number of potassium will leave the cell comparison to sodium. The rate is faster in case of potassium as compared to sodium. Okay, and for resting potential value, if we talk, it will become around say minus 70 MV. This is your resting potential value. Okay, so in depolarization, the reverse case will occur. That means potassium will move out and sodium will goes in, which will change the charge from negative to positive. This is the vice versa case of polarization stage. Now we can say that the cell is depolarized, and this position is known as depolarization or action potential. Action potential. Got it? This is just the reverse case of polarization, where the cell acquires a positive charge by releasing potassium and by accepting sodium. Clear? Now we'll be discussing the third one. In third, what will happen? See, now let's have a clear idea. This is the resting potential and this is the action potential. So what happened in this two cases? Let's suppose here is a negative charge, this is your positive charge. Here potassium concentration is high, sodium concentration is low. But here potassium concentration is low and sodium concentration is high. That means in resting potential, the cell is permeable to potassium and impermeable to sodium. That means here in action potential, the cell will be permeable for sodium and impermeable for potassium. Now it is very much essential to attain the original position, because again we have to go back to the original position. That means the action potential will come back to the original position by closing the sodium and potassium pump. That means whenever the cell will attain the maximum concentration for sodium and minimum concentration for potassium, the potassium sodium pump will automatically closed, which can transfer the depolarized state into again polarized state. This state is known as repolarization. So this is your first step, polarization resting potential. This is your second phase, which is known as action potential, and this is your third step, which is known as repolarization. By this process, the electrical fields or the impulse or the signals can transfer from one cell to other cells in a continuous manner. So this is your mechanism for neural transmission. This is done through the help of cleft. The cleft is between dendrites and axon terminal. These kinds of information can be done here. So this is all about the structure of neurons and the mechanism and functions of different types of neurons.